This lecture will cover the basics of synaptic transmission in the nervous system. The nervous system has billions of neurons, and each of them can have thousands of contacts. These neurons are in constant communication, even when we're sleeping, so the sheer amount of activity is truly mind-boggling. Despite this, neuronal communication is made possible by a couple of different types of electrical signals, which are linked by chemical messengers. Let's zoom in for a closer look. Neurons have two important jobs. One is to transmit a message to a target neuron across a synapse, using neurotransmitters as messengers. The type of electrical signal resulting from this is called a graded potential. If the graded potential is the right size and type, then it allows the neuron to do its second important job to carry the message along its length to its target, using a type of electrical signal called an action potential. When an action potential reaches the terminal of the neuron, it triggers neurotransmitter release, which produces a graded potential in the target neuron, which can then produce an action potential in this neuron, and so on. This continuous cycle of communication is running all the time in your nervous system. For this tricky topic, we'll focus on the events at the synapse and learn how synaptic transmission plays a role in neuronal communication. We're going to start our journey to the synapse with a bird's eye view. The terminals of the neuron on the left, shown here in red, form synapses with the dendrites of the neuron on the right. Although most neuronal terminal synapses on dendrites, like you see here, keep in mind they can also form synapses on other parts of the target neuron. The sending neuron on the left is called the presynaptic neuron, and the receiving neuron on the right is referred to as the postsynaptic neuron. Keep in mind that these are relative terms like left and right. Most neurons act as senders and receivers of information, so they're both presynaptic and postsynaptic depending on which event we're referring to. If we pick this particular synapse right here, we'll have to zoom in to see the detail. Synaptic transmission starts when neurotransmitter released from a presynaptic neuron binds to its receptor. At this magnification, we can see little bubbles called synaptic vesicles, and inside these you can see neurotransmitter molecules. This vesicle here is releasing its neurotransmitter into the synaptic cleft, and it can travel the short distance to the postsynaptic neuron here. The synapse is tiny, so let's zoom in for an even closer look. The liberated neurotransmitter is able to travel in the small space of the synaptic cleft, and once it binds to the receptor on the postsynaptic side, the message then gets passed to the next neuron. There are lots of different types of receptors, but the simplest ones are gated to ion channels, like shown here. Once a neurotransmitter binds, it opens the ion channel part of the receptor, allowing certain ions to travel across the neuronal membrane along their electrochemical gradient. Note here that only neurotransmitters or drugs that fit the receptor site are able to open the ion channel. Let's review the steps so far. First, neurotransmitter is released from the presynaptic terminal. Then, neurotransmitter binding to the receptor opens up the ion channel on the postsynaptic side. What happens next in the postsynaptic neuron depends on the type of receptor and its ion channel, since this determines the type of graded potential generated. If the channel opens and the inside of the neuron becomes less negative than the resting potential of -70, this is referred to as a depolarization and is considered excitatory. If instead the neuron becomes more negative than -70, this is referred to as a hyperpolarization and is considered inhibitory. Let's look at some examples of different types of receptors which produce different types of graded potentials. First, let's look at depolarization. A neurotransmitter that is always excitatory at its synapses is glutamate. So we can pretend that the green diamonds are glutamate molecules and the pink circles in the synapse are sodium ions. When the glutamate binds to its receptor, as shown in the middle panel, the channel opens and the sodium ions are driven by the electrochemical gradient. That's the difference in concentration of sodium and the difference in charge on each side of the membrane. So at rest, we can measure a voltage of -70 millivolts, but when sodium moves into the cell through the receptor ion channel, it brings with it its positive charge, making the cell more positive, which is called a depolarization. If the channel remains open for longer, more sodium will flow in, making the cell even more positive. This type of graded potential is called an excitatory postsynaptic potential, or EPSP for short. What about hyperpolarization? A neurotransmitter that's always inhibitory at its synapses is GABA, and its receptor channel is permeable to negatively charged chloride ions, shown here in blue. When GABA binds to the receptor, as shown in the middle panel, the channel opens and chloride ions are driven into the postsynaptic neuron because of the concentration difference. There's more chloride outside of the neuron than inside. So at rest, we can measure a voltage of minus 70 millivolts, but when chloride moves into the cell through the receptor ion channel, it brings its negative charge with it, making the cell more negative, which is called a hyperpolarization. If the channel remains open for longer, more chloride will flow in, making the cell even more negative. This type of graded potential is called an inhibitory postsynaptic potential, or IPSP for short. So why does this matter? Well, if the postsynaptic neuron adds up all of the EPSPs and IPSPs, and the membrane potential reaches minus 55, a different type of ion channel comes into play, one that's opened by a change in voltage of the neuron. The ion channels we've looked at so far are opened by neurotransmitter binding. But these voltage-dependent sodium channels are sensitive to changes in charge. When these sodium channels open, it creates an action potential, which allows this neuron to carry the message to its target. Overall, synaptic transmission may seem quite tricky, but it's not too complicated once you know the basics.